Hello. Yes.
Switch on your camera and
Just 1 minute. Is it visible?
it is visible also in the screen, no problem. What we now
Okay.
need is the sound. Now the sound. यहाँ से sound देखिए. The problem is that the sound is very little.
Okay. So below there is a microphone sign, and if you go to a small arrow, there are some option like default external microphone.
Below there is a mic.
Is it okay? Can start.
इसका sound over नहीं कुछ. बैठो बैठो यहाँ बैठो. बैठ गया बैठो. चार्जिंग को भी लगानी पड़ेगी. चार्जिंग ले चार्जर ले. Start, start speaking.
Okay, we can start now.
करो.
Okay, so I'm sharing my screen.
Right now, this
Ah,
is like 10:50 already. So these
okay.
students go for a class at around 11:20. Huh?
Oh my God. Okay.
So we have
Okay.
half an hour. Can we give
Okay.
a break then and come back if you can?
What time?
They will be back by 12, 12:15. 12:15 to
Um, 12:15. It will be difficult actually. I have some other meetings also.
okay.
Uh,
Second half, second
second
half,
half I can. Second half I can try maybe little late around 2:33 is okay.
three.
Three, three is okay.
हाँ वो हो जाएगा हम लोग कुछ कर लेंगे. हाँ? ठीक है. सो वो तो उनको बोल के थ्री तक वो लोग चला देंगे. वो एक बार बात कर लेंगे. ठीक है. सो
Okay.
उठाइए सर. अभी कंटिन्यू करो टिल 12:50.
Okay.
हाँ, because most of the students
Okay.
have to go because we are already late. So then
Okay.
maybe another half an hour or so, right?
3 o'clock. Okay, fine. So this same meeting link will work, or you will send a new link?
That we will let you know.
Okay, fine. So my screen is visible.
Start.
Okay. So good morning, everyone. Before we start, like I hope you have little bit idea about microscopy, and if not, then we can actually start from basics actually. And
Can you go?
today, huh?
Yeah.
Okay, so today's topic will be type of microscope and then how the optical microscope generally work and what are the different type of bright field contrast technique. Then we'll go to fluorescence microscopy and confocal microscopy, and finally I'll talk about little bit very briefly about what it means by resolution limit of a optical system, and then very superficially we'll touch about super resolution microscopy. So what we can do, if time permits now, we can cover the point number 1, 2, 3 now, and probably from 3 o'clock we can start from fluorescence to like super resolution. Okay, so if we try to understand what is microscope, so this term is actually taken from a Greek word. So micros means small, and scoping means to look or to see. So that means microscopy means like some technique of some instrument that we use for seeing a very tiny object which is not possible to see with a normal eye. Okay, and so that is actually the name actually come from. And if we very grossly divide the microscopy, so there are two different type of microscopy technique, like a type on based on the illumination type we can divide them in optical microscopy, where we use light photon or light particles for illuminating the sample. We generally use visible or IR light, and there are some other options also. And there is a very common electron microscopy where you use electron beam for illuminating our sample. So these are the very grossly two different type of microscopy. And if we talk about microscopy structure, based on the structural complexity, we can divide in two different groups. One is simple microscope. It's very older, like when the microscopy just started, that time people generally use simple microscope, but nowadays mostly all the microscope are compound microscope. So sorry. So in case so when we talk about simple microscope, it has only single lens. Okay, so we have this object, probably you can see my cursor, or I can go to laser pointer maybe. So you can see this is the object, and with the lens actually we can magnify the object and we can see the image. So this one example of simple microscope is that magnifier glass or something like that. And if we talk about compound microscope, there are generally two different type of lens. There might be some intermediate lenses also. One is eyepiece tube, another is objective lens. When they come together, then only it magnify the image even more. And you can see like this is the object, and with the eyepiece actually we have first image formation here, and after with the objective lens, finally we got this magnified image, which is the lower red arrow. So that is how, with the help of objective lens and eyepiece lens, we can even magnify more for the object. And this is light path of the how the image is formed. So as I shown you in the last slide also, this is the same. And based on that, actually there are some length like focal length and other things. I'm not going into the details, but as you can see, this is the object actually, and this is the final image that is formed. And if we have a let's say eyepiece tube of 10x magnification and objective of like 100x magnification, so there will be 10 into 100, that means 1000 times zoom. Okay, 1000 time magnified object we can see with the microscope. And if we talk about the structure of the microscope, there are two different type of structure. One is inverted microscope, another is upright microscope. So in case of inverted microscope, we have the objective at the lower position, and above that we have the stage where we can put the sample. So inverted microscope is mainly used for normal day-to-day imaging with a fixed sample. And also you can use the inverted microscope for live cell imaging, like where you use some particular dish or some cell culture plate to see your sample. But there is another type of microscope where which is called upright microscope. In case of upright microscope, you can see the object on the objective on the upper side and the stage on the lower side. So you place the sample and the light is coming from the top. So this upright microscope is mainly used by the clinicians because it is very easy to use and easy to change the sample. So that is why people, particularly who are working on clinical samples or medical field, they actually use the upright microscope. So these are the two different type of microscope, and there are different type of application that it can cover. Based on that also, you can decide which microscope you should go with for your particular application. And this is very important part. You when you want to understand the light path of the microscope, there are two different type of light path. One is called transmitted light path or diascopic light path. Another is called reflected light path or episcopic light path. So you can see this is the upright micro sorry, this is an inverted microscope. On that, this is the lamp house. From this lamp house, there will be a LED or a halogen light, which is creating the bright field image. So light is coming this way, and then through the condenser lens, it is passing through the sample, and from there it is finally going to your eyepiece, or you can send to a camera for imaging. So here, actually, the light. This is a sample which is placed on the stage, and the light is actually passing uh, through your sample. So that is called transmitted light path or diascopic light path. And on the right side, you can see there is a reflected light path that is mainly used for fluorescence observation. So transmitted light path mainly used for the bright field observation normally, but there is also variation. But in case of reflected light path, that is mainly used for fluorescence light source. So you can see this is the light source, and from this light source, light is coming, and there is a mirror which actually change the light path and bring the light inside to the microscope body. And here we have different filter cubes. I will come to that, what are filter cubes and about the details. And from this filter cube there is a dichroic mirror that sends the light to your sample, and whatever excitation and emission, that emission is coming back. It's like a reflection, like light is going, excitation light is going to a sample, and then emission is coming back. It's like a reflection, and then finally you can see the emission with the eye, or you can do imaging with the camera. So that's why it's called reflected light path or episcopic light path. So these are the two light path that is mostly used for a microscope technique, optical microscope technique, and you have to understand this light path very clearly to understand all the optical components and how you can use this light path for your imaging application. So when we talk about optical microscopy, generally we use a whole, we can use the whole spectrum, which is like from 400 to 700, which is our visible spectrum, or we can use a particular area or particular bandwidth of light for excitation of the fluorescence that we use generally for our fluorescence imaging. So when we see our sample, let's say if we want to see our sample under normal bright field technique, so there are some bright field technique which is called phase contrast microscopy, DIC or differential interference contrast microscopy, and then we have the dark field microscopy. So these are the three techniques based on bright field light path. And then we have fluorescence microscopy, which is based on episcopic light path, confocal same, and again super resolution same light path. So the first three techniques are for the contrast technique based on the bright field imaging. Now, why we need a bright field contrast technique? So if we concentrate on the left side of the image, you can see, we can see the cell, but the boundary of the cell, it's very difficult to understand in with a normal eye observation. So now, to understand the cell boundary properly, or sometimes people also want to see the internal little bit structural details uh, uh, of an unstained sample where we are not using any dye. So under normal bright field, we want to see internal details of the cell. So that time it is good to go with a phase contrast technique. So this phase contrast microscopy, it's a contrast, like enhancing the contrast in the bright field. So that is first actually introduced in 1934. And actually there are some optical components through which you create a difference in the phase of the light rays. And since there is a change or difference in phase, and that is finally creating the phase contrast effect. So I'm not going into the all the optical details, just please remember these two three things. One is left side, you can see the bright field bright field image, and on the right side you can see the phase contrast image. And for a phase contrast microscopy, there are two components that is required. So one is called phase annulus, and another called condenser annulus or condenser ring. So the phase annulus is placed in like before the objective. There is a condenser turret where we can put that ring, and inside also, in the inside the objective also, there is a ring. And when they two come in the optical path, then only this phase effect is created. So you can see, so these are upright platform, so light is coming this way, and we put condenser annulus somewhere here below the objective, and we put the phase ring here, and when they come together, that is creating the phase contrast effect. So if you compare on the left side and right side image, you can see left side is bright field image, where it is very difficult to understand the hair like projection, but on the phase contrast effect, it's very easy to understand the hair like projection structure. So that is how the phase contrast technique can help you to see internal details of a unstained sample. So as usual, every technique has some advantage and disadvantage. So advantage is that you can see the unstained sample with internal details, but the disadvantage is that since you are using the phase ring and condenser annulus, so when it is actually cutting a lot of amount of light, so as a result, and it will also decrease the resolution of the image. Okay, and sometimes in the phase image, you can see there is a hollow kind of effect that is actually very common to phase contrast image. Now there are some advancement in case of phase contrast image with respect to the objective to reduce the halo effect. Okay, so this is the phase contrast technique. And now we will come to the DIC. DIC is mainly required. So if I want to see the surface of the cell, because for my, let's say for my application, I am interested to see the surface of the cell, but not the internal structure of the cell. So that time you can always go with a DIC microscope, and DIC actually will give you some 3D kind of impression of the cell surface. And from this, you can understand the cellular surface morphology, not morphology, that cellular surface details more clearly. So on the upper part, you can see this is a DIC image, and you can see the like it's like a pseudo 3D effect that will be be the advantage of the DIC image. And since we use the full illumination or full aperture of the light, so DIC image has better resolution compared to phase contrast. So on the left side, you can see, like if we see the back aperture plane of the objective, at the left side you can see this is actually filling up the whole back aperture of the objective. So as a result, this will give you better resolution compared to on the right side, which is for the phase contrast. But here the disadvantage is that we can't understand the internal structure that clearly, which is possible with the phase contrast. So that is the difference between the DIC and phase contrast. And based on your requirement, you can choose any of the contrast technique, and you have to put that optical components into the light path. Then only that kind of effect will be created. So for DIC, we need four optical components. One is called analyzer, and this is called polarizer, and two prisms. And when these four components come into the optical path, then only this DIC effect will be created. So one analyzer or sorry, so you can see this is the analyzer, and before that objective we have one prism which is called Nomadsky prism, or sometimes it is called Wollaston prism also. And then we have another prism here, and finally we have the polarizer here. So when analyzer and these two prism and finally polarizer come into the optical path, then only that DIC effect is created. If you take out any of the component, then probably you can see the DIC effect is much reduced, or there is no DIC effect. It's like a normal bright field image. Okay, so that is why you need to have all the four components in the optical path for this DIC effect. And finally, there is another optical contrast technique which is called dark field microscopy. So in case of dark field microscopy, you can see what we do. We actually, from this light source, whatever light is coming from the condenser lens, central part is blocked. Only the light coming from the very close to the periphery of the condenser lens, that is allowed. And then whatever light is traveling through the sample, that got diffracted. And here also we block the central part of the light, and only the peripheral light which is coming due to the diffraction, that is actually used for your observation or imaging. And as a result, what happens, like lot of light got cut or blocked in the light path. That is why we need to have a dark for dark field microscopy, we need to have a very bright light source that will compensate that loss of light. And when that light actually if we see a sample under that dark field condition, you can see the background of the sample, like the outer part of the sample is totally dark, and you can see the sample details very clearly, the projection or some other things, it's very clearly you can see. So sometimes that dark field microscopy is used for some specific application. And here the advantage is that the background is totally dark, so you can see the finer details, or the contrast is good. But the disadvantage is that since you are blocking a too much amount of light, so you have to use a very high light source or high intensity light source, and that sometimes can create a problem for your particular sample, which is not very friendly condition, because sometimes sample does not like it is not able to tolerate that high intensity of light. So that is why that is the disadvantage of a dark field microscopy. So as I told you already, that based on your application requirement, you have to select which kind of microscopy technique you should go with. And finally, if we see the same sample, so let's say we take some tissue paper, the fiber of the tissue paper, and if we see under three different type of contrast technique, you can see this is left side is a dark field where you can see the background is totally dark and we can see the fibers, and then normal bright field image, and right side it's a phase contrast image. Okay, so the same sample will appear different in different contrast technique. So that is how the bright field contrast technique is helpful, particularly for our different application. So is there any question, or should we continue the fluorescence microscopy? We can do now. Hello.
Yes.
Is there any question or anything we can discuss, or we can like we have 8 minutes left till we can go with the fluorescence microscope. We can go to fluorescence. Okay. So, do you have any idea about what is meant by fluorescence before I go to fluorescence microscopy? Anyone has studied what is fluorescence, what is fluorochrome? Any idea? Okay. Okay, fine. So, what I want to tell you, let me go to this. You can see my screen. Okay, so you can see there are some chemical molecules or some protein molecules also that actually give you the fluorescence. What is mean by fluorescence? So let's say like if you have a particular molecule, that kind of molecule, which is called fluorophores or sometimes fluorochrome or fluorescence proteins. If you put a particular wavelength of light, that will go to high energy state, and from that high energy state again it will come back to the ground state, and when it comes to ground state it actually emits some particular wavelength. Okay, so the light which is specific in wavelength light which is used for excitation, that is called excitation light or excitation wavelength, and whatever light is actually emitting by the fluorophore molecules, which is called emission light or emission wavelength. Now, when we talk about how the molecule can go from ground state to upper excited state, that can be done in two different ways. One is like a single photon or light particle can transfer the energy to that particular fluorophore molecule, and it can go to a high energy state. Or sometimes we use like low energy photon or low energy light, but they that time we need, let's say, two number of photons to transfer the energy to bring the molecule to the upper excited state. Okay, so left side, when there is single photon transferring the whole energy to the fluorescence molecule, it's called single photon excitation. And on the right side, you can see there are two photons actually transferring the energy, and that is called two photon excitation. So I don't know whether you have heard about confocal microscopy, multiphoton microscopy. If you go little details like how that light source actually help in microscopy excitation, so you can see if normal day-to-day imaging like fluorescence or like confocal, we use generally single photon excitation or visible light. But when we talk about multiphoton microscopy, we use two different photon, two photon excitation, which is actually lower energy and longer wavelength range. And why this is important? Because let's say sometime people wants to see what is happening inside the mouse brain. Now, if you want to image inside the mouse brain, you have to go deeper into the tissue or into the mouse brain. And to go into the deeper section of the mouse brain, you need a longer wavelength of light because the penetration depth are better compared to the visible wavelength range. So that is why two photon excitation people generally use for multiphoton microscopy, and multiphoton microscopy generally used for situation where you want to go deeper inside the tissue or the sample. So these are the two different particular excitation profile that we have to understand. Now for the fluorescence light, actually there are different light source. Initially, like earlier, we used to have this mercury illumination. We have this mercury bulb or xenon bulb that we use for mercury light source, which has the all the wavelength range mostly. And nowadays, people actually shifting to LED based fluorescence. So here, the LED light source can have specific LED with specific wavelength range, or it can have a white LED to emit the all the wavelength range. Okay, so there are options available in the market from different manufacturer, and based on your application, you can select. Now, why people now changing from mercury to LED? Because mercury light source, it has a very like low lifespan. It is around, let's say, 2000 hours. But if you want with the LED, it is around, let's say, around 15 to 20,000 hours. So almost 10 times more. And also the LEDs are very stable. So like what happened, like after 2000 hours or near to 2000 hours, you can see the mercury illumination is going down. So the excitation capability will go down, and also sometime it goes off. There is a flickering. Okay, but with the LED, it's very stable and throughout the lifespan, and you can actually quickly switch on or switch off the LED light source, which is not possible with the mercury. If you switch on, you have to wait for some time, and when you switch off, you have to wait for cooling down. And once it cool down, it takes around 20 to 30 minutes. After that only you can again switch on. So that is why people actually going from mercury to LED based fluorescence illumination. Now, when we talk about fluorescence microscopy, let's say we are using a mercury illumination. So all the light is coming to your microscope body. Now, what you have to do, you have to select a particular range of the light which you can use to excite a particular fluorophore set. So again, I just want to know, have you heard of any fluorophore molecule? Any fluorophore molecule you have heard, like DAPI, FITC? Okay, so there are fluorophore molecule. It's called DAPI. DAPI probably you will use when you do the lab. DAPI is actually going inside the nucleus, the double-stranded DNA, and bind there. So it actually, when you stain or you incubate your sample or cell with the DAPI, it will particularly stain the nucleus. Okay, and there are some particular small molecule. Probably you have heard about actin fibers. Actin fibers you have heard, actin microtubule. Okay, so actin fibers actually, there are some small molecule which is called phalloidin. Phalloidin binds specifically to actin fibers. If you put inside a cell, it will go inside of the cell and bind to specifically the actin fibers, no other other molecules. Now if you tag the that phalloidin with some fluorophore molecule, let's say green fluorescence, which is called FITC or Alexa fluor 488. Now what will happen if you excite wherever you have the actin fibers that is already bound with this small molecule phalloidin, and with the phalloidin you have a fluorophore. So from the excitation emission you can understand where is the location of the actin fibers. So that is how inside a cell you can understand what is the location of the nucleus, what is the location of the actin fibers, and there is another small molecule which is called mitotracker. So mitotracker actually binds mitochondria very specifically. It does not bind to lysosome or Golgi body or endothelium ER particularly. So it binds only to mitochondria. So if you incubate with a mitotracker red, or sometimes there is other version also, you can see the mitochondria under the red color or red fluorescence light. Okay, so that is how if you put different molecule, you can stain particularly a specific structure inside the cell, and you can understand the location of that particular structure. So let's say for your particular application, you need to understand whether there is a ROS generation or reactive oxygen species generation inside a mitochondria, and how the mitochondria get fragmented. So what you can do, you can have a control and you can have a treated sample, and you can add mitotracker rate, and you can see the two sample under fluorescence, and probably in the control sample you can see elongated normal mitochondria, Wherever in the treated sample, you can see mitochondria become very small globules, round color type of structures. So that means mitochondria got fragmented. From the image observation, you can actually tell or you can conclude, okay, my mitochondria has some damage, or this reagent is causing some damage. So that kind of interference or conclusion you can draw. Okay, so should we stop now? You have something, some class maybe? We can start from 3 o'clock. Is it okay? Okay, fine.